Welcome to G Fun Facts Online. When you think of the most colorful animals on Earth, you probably picture a peacock's shimmering tail, a tropical fish darting through a reef, or a jewel beetle with its metallic carapace. Mammals, on the other hand, tend to be a bit, well, brown, gray, and black. But hiding within this seemingly muted class of animals is a secret language of color, one that's not written with pigments like melanin, but is built with microscopic architecture. This is the realm of structural color, and while it's exceptionally rare in our furry relatives, the few examples that exist tell some of the most fascinating stories in all of evolution, a tale of physics, behavior, and deep time. First, let's draw a clear line between the two ways nature makes color. Most color you see, from a red apple to your own hair, comes from pigments. These are molecules that create color by absorbing certain wavelengths of light and reflecting others back to your eye. Pigmentation is a subtractive process. It takes away parts of the light spectrum. Structural color is completely different. It's an additive process born from physics. It's created when light interacts with nanoscopic structures, surfaces so incredibly tiny they're on the scale of light waves themselves, typically between 400 and 700 nanometers. These structures, often made of completely transparent materials like keratin or collagen, act like photonic crystals or diffraction gratings. They sculpt light, scattering it, and causing light waves to interfere with one another. When waves interfere constructively, that color is amplified and becomes brilliantly visible. This effect was first noted by pioneers like Robert Hooke and Isaac Newton in the 17th century when looking at peacock feathers, but it was Thomas Young in the early 19th century who explained the principle of wave interference, giving us the physical foundation for how these structures work. A key hallmark of structural color is often iridescence. The color changes depending on your viewing angle. Think of the shifting rainbow on a soap bubble. That's because changing the angle changes the path light travels, altering which wavelengths are amplified. So why is this dazzling effect so vanishingly rare in mammals? The leading hypothesis is the nocturnal bottleneck. For about 150 million years, while dinosaurs dominated the daytime world, our ancient mammalian ancestors were largely small, nocturnal or crepuscular creatures, active at dawn and dusk. In a world of low light, complex visual signals are far less effective than other senses like smell and hearing. There was simply no evolutionary pressure to develop or maintain vibrant color displays that no one could see. This long period spent living in the dark had a profound and lasting impact on mammalian vision. While early vertebrates were likely tetrachromatic, with 4 types of color-sensing cone cells, most mammals today are dichromats. We lost 2 of those cone types, giving us color perception roughly analogous to red-green colorblindness in humans. For most of their history, most mammals couldn't have even perceived the brilliant structural colors that adorn other animal groups, making their evolution a non-starter. On top of that, hair itself, which is a defining feature of mammals, primarily evolved for insulation and touch, and its basic structure may not be as easily modified into the precise, periodic nanostructures required for iridescence as bird feathers or insect cuticles are. But evolution is never static. When some mammals moved back into the daylight, and especially when some primates re-evolved more complex vision, the stage was set for color to make a comeback. Perhaps the most paradoxical and beautiful example is a creature that can't even see its own splendor, the golden mole of southern Africa. These small, subterranean insectivores are functionally blind. Their eyes are vestigial and covered by skin. And yet, their dense fur shimmers with a stunning metallic or iridescent sheen of green, blue, violet, or copper. This immediately rules out typical drivers for such color, like attracting a mate or recognizing species. So why would a blind creature living in perpetual darkness evolve an iridescent coat? The answer is a classic case of an evolutionary epiphenomenon, where a trait arises as a byproduct of selection for a completely different function. Detailed microscopic investigation revealed the secret. The iridescent hairs of the golden mole are flattened, not round like most mammal hair. Furthermore, their cuticular scales are dramatically reduced and compressed into smooth, overlapping layers. This structure acts as a multi-layer thin film reflector. As light passes through these transparent layers, it reflects off the top and bottom of each one. Certain wavelengths interfere constructively, amplifying them into a visible color, while others cancel out. The real reason for this structure has nothing to do with color. The flattened, smooth hair profile is hydraulically and aerodynamically superior for moving through a dense medium. For the golden mole, which essentially swims through sand and soil, these specialized hairs reduce friction and resist abrasion. This allows the mole to move more efficiently, saving precious energy. The robust, layered structure also likely makes the fur more durable and better at repelling moisture and dirt, essential for an animal that spends its life underground. The iridescent color, then, is not the trait that was selected for. It's simply a coincidental and beautiful physical consequence of evolving a coat perfect for burrowing. In stark contrast, we have the brilliant blues displayed by certain primates and marsupials. This is not an accident. It is a deliberate and powerful form of visual communication. The most famous example is the male mandrill. Charles Darwin himself was so struck by it that he stated, "No other member of the whole class of mammals is colored in so extraordinary a manner." The vivid blue coloration is most prominent on the face and hindquarters of male mandrills, forming a stark, unmissable contrast with the vibrant red of their nose. Similar, though less dramatic, blue skin is found on the scrotum of vervet monkeys and, remarkably, in 2 species of opossums. The evolution of this trait in both Old World primates and marsupials, groups that independently evolved trichromatic vision, is a stunning example of convergent evolution. For over a century, this blue was incorrectly attributed to Tyndall or Rayleigh scattering, the same phenomenon that makes the sky appear blue. But detailed studies using electron microscopy revealed a far more ordered and sophisticated mechanism. The blue color is produced by coherent scattering from quasi-ordered arrays of parallel collagen fibers in the dermis, the layer of skin beneath the epidermis. These collagen fibers, each about 100 nanometers in diameter, are arranged in a dense, semi-regular pattern that functions like a 2-dimensional photonic crystal. This structure is perfectly tuned to selectively scatter and reflect blue light back towards the observer through constructive interference, while other wavelengths pass through and are absorbed by a layer of dark melanin pigment located just beneath the collagen array. Because the array is quasi-ordered, meaning it has short-range order but lacks a perfect repeating lattice, the color is non-iridescent. It appears blue from all angles. The intensity of this blue is directly tied to a male mandrill's social status. The development and maintenance of this color are physiologically linked to hormone levels, particularly testosterone. A dominant male has higher testosterone, which not only influences the red coloration by increasing blood flow, but is also associated with the development of more ordered and dense collagen arrays, producing a brilliant, saturated blue. This makes their colorful face an honest signal of fitness and dominance. It reliably communicates his status to both rivals and potential mates. It's a billboard that advertises strength and genetic quality, deterring challenges and attracting females. Finally, we come to the most widespread yet most hidden example of structural color in mammals, the tapetum lucidum. If you've ever shone a light into the eyes of a cat, dog, or deer in the dark and seen their eyes glow with an eerie light, you've witnessed the tapetum lucidum. This bright tapestry is a layer of tissue located immediately behind the retina. Its function is to act as a biological light amplifier, a retroreflector. Light entering the eye passes through the retina, where it has one chance to be absorbed by photoreceptor cells. In animals without a tapetum, like humans, any unabsorbed light is lost. But the tapetum reflects that light back through the retina, giving the photoreceptors a second chance to capture the photons. This mechanism can increase the light available to the photoreceptors by as much as 50%, granting superior night vision. The eyeshine itself is a form of iridescence, and its color, blue, green, yellow, or red, depends on the reflective materials and structure. The tapetum lucidum is another remarkable example of convergent evolution. Mammalian tapeta fall into two main types. The first, the tapetum cellulosum, is found in carnivores like cats and dogs. It consists of layers of cells containing organized, highly refractive crystals. In cats, it's crystalline riboflavin. In dogs, it's a zinc-cysteine complex. These crystals are arranged in precise layers that function as a thin film interference reflector, far more efficient than the golden mole's fur. A cat's tapetum is estimated to be 130 times more reflective than the back of a human eye. The second type, the choroidal tapetum fibrosum, is found in herbivores like cows and horses. It is not cellular, but is composed of an array of precisely arranged extracellular collagen fibers, functioning via coherent scattering, much like the blue skin of mandrills. The study of structural color in nature is not just an academic exercise. It's a frontier of inspiration for new technologies. The field of biomimicry seeks to emulate nature's time-tested designs, and structural color offers a wealth of possibilities. Unlike pigments and dyes, which can fade with UV exposure and often rely on toxic heavy metals, structural colors are incredibly durable and can be produced from abundant non-toxic materials. Imagine paints that never fade, cosmetics that produce vibrant hues without synthetic dyes, or textiles that shimmer with effects woven into their very fabric. The principles seen in mammals offer unique insights. The quasi-ordered collagen in mandrill skin provides a blueprint for creating stable, non-iridescent structural colors, which are highly desirable for coatings and displays. The golden mole's hair, optimized for reducing friction, could inspire durable, self-cleaning surfaces for burrowing robots or low-drag coatings for vehicles. And the highly efficient retro-reflecting properties of the tapetum lucidum could inform the design of novel optical sensors, low-light imaging systems, or reflective materials for safety applications. In conclusion, the world of mammalian structural color, though subtle and rare, offers a profound perspective on the ingenuity and opportunism of evolution. It reveals a story written not in ink, but in architecture. From the accidental shimmer of a blind mole's fur, born from adaptations to a life in the dark, to the calculated, vibrant blue of a mandrill's face, a testament to the power of social competition in the light, we see how the same physical principles can be deployed for vastly different evolutionary ends. The tapetum lucidum, a hidden iridescence, showcases convergence on a grand scale as different lineages independently fashioned biological mirrors to conquer the night. These examples demonstrate that the mammalian evolutionary journey, while long dominated by the muted tones of a nocturnal past, retained a latent capacity for optical brilliance. In studying these shimmering enigmas, we not only gain a deeper appreciation for the diversity of life, but also unlock a new palette of possibilities for our own technological future. The iridescent mammals stands as a testament to the fact that in the intricate dance between physics and life, evolution is the ultimate nanotechnologist. Thanks for tuning in to G Fun Facts Online.